Thank you for joining us. My name is Dishen Kumar, and today we have a very interesting topic. We're actually talking about DNA, our genes, and actually the essence of life. So today with me my guest is Professor Dr. Zafarina Zainuddin, the Director for Analytical Biochemistry Research Center for USM. Thank you, Prof, for joining us today.
Thank you for having me here.
So, Prof, the first thing that came to me when I was looking over my notes and things like that, the first question was, what is actually DNA ancestry?
Okay, so ancestry is actually based on one study using DNA mitochondria. This DNA is very unique to human being because it is only inherited from the mother line. Saying that means your DNA can be traced to your great-great-great-grandmother, so you will have the same mitochondrial DNA profile as your great-great-grandmother. So one of the biggest thing in this mitochondrial study started around 1970s, where they came out with this theory saying that as an out of Africa, saying that our modern human actually traveled out from Africa around 70,000 years ago, and then this group of people actually traveled south to the land called Sundaland. Previously, it was
Southeast Asia.
Southeast Asia, and then another group traveled north to the Europe and everywhere to the north side of the world. And the special thing about this study, they found that the oldest or the most recent common ancestor of this modern human, was actually found in Africa, and that particular DNA type was called mitochondria Eve, and this actually DNA was actually dated about 200,000 years ago.
Oh wow, 200,000 years ago.
Yeah.
Okay, and speaking about all that, it sounds so interesting, getting to know where you're coming from, where we actually started, where life started and things like that. How did you actually start in this? What is your story?
Okay. When I did my PhD in University of Glasgow, first actually I started with a very quite complicated proposal. I wanted to study on this disease called syndrome infant death.
Okay.
So also known as the baby cot death, something like that.
Okay.
But my problem was to get sample was very difficult. So, and during that time, my supervisor was actually analyzing this Neanderthal's DNA in our lab. So he did ask me, do you have any aborigines group in Malaysia that are very interesting to study, that we can relate to this out of Africa theory? And that was the time I was like, yeah, we do have these Negrito people, so they might be actually one of the descendants of the oldest population in the world. So I came back to Malaysia and then did some background check and everything. So I found these Semang groups. They were actually being cited somewhere, saying that they are actually among the relic population, and I think they are worth to study that group. So what I did was actually to compare this orang asli DNA to the Malays, and that is where I actually started to get this interesting finding, where we can find some of the orang asli DNA that are very old, up to about 60,000 years ago, that was also found in the Malay DNA.
Okay.
So when I wrap up my study, I submitted my thesis. The first thing that I did when I came back to Malaysia was to apply a grant, and then I studied I continued the study using mitochondrial DNA for the Malay population, but we are actually narrowing down into the subgroups, Malay sub-ethnic groups, and we did some comparison with the Chinese, with the Indians, and there we can find that the haplogroups for Malays are very specific to Malays, and the haplogroup for Chinese can be traced back to the China mainland, and the Indians, of course, to the Indian continents.
And speaking about that, maybe we can talk about a bit more. You were saying the Malays have a different type of mitochondrial DNA, the Chinese and the Indians and things like that. Is this a very big difference, or is this just to show that we actually came from different parts of the world at a specific time and things like that?
Yeah. Okay, so let's say if I'm talking about the Chinese, yeah. So the Chinese and the Malay and the Indian, we are actually comprised in one big haplogroup known as the M haplogroup. And we have these Europeans, they are more into these R and N haplogroups. So these haplogroups, these major haplogroups, macro haplogroups, are continent specifics.
Oh, okay.
All right. But in the M, we have ME, MD, MC. These MD, MC, M21 and everything, they are ethnic specific. So by looking at narrowing down the search, by looking at more sequences from the mitochondrial DNA, we can actually cluster them in a smaller groups. But again, I can actually have a big group of Southeast Asian, I can have big groups of East Asian. So these are the differences between, that we can say that small differences between the races.
And speaking about that, the question of race. Now, if we talk about DNA ancestry and finding out where people come from, things like that, aren't we kind of, one questions be actually asked about compartmentalizing people into boxes or into races, and does that actually divide us more than unite us?
Putting us together.
Yeah.
All right, okay. First of all, when I started my study, it was actually, my specialty was actually in DNA profiling. And the use is actually in crime investigation.
Oh, okay.
So when we use DNA in crime investigation, when we get a DNA sample or profile from a crime scene, and when we try to actually compare to the suspect, it's not actually to find him guilty, but it's more into to exclude him from the case. So when a suspect is not guilty, so it will be much easier for them to actually to carry on with their lives, right? So that is the main role of DNA in crime. So same goes like this one. We are actually looking, the good thing is actually for us to understand where do we come from, so are we actually related to each other. So let's say myself, when I did my PhD, I did on my DNA.
Oh, okay.
So interestingly, during that time, early 2000, I couldn't find where I came from. So after a few years later, when my PhD student started to actually test more people and the database is growing, so then only I can actually, my DNA type, mitochondrial DNA type was actually very specific to Papua New Guinea.
Oh, wow.
So then only I know how my great-great-grandparent, my great-great-grandmother traveled as far as from Papua New Guinea to the Java, because my great-mother was actually from Java, and then up to Johor, and there is where my great-grandmother family started. So the good things are there, and if you want to actually look at the negative point, people
Anything,
can just
yeah. Negatively, okay.
Yeah.
Talking about that, if a person does a DNA ancestry, what information do they actually get?
Okay. For DNA ancestry, a very specific study, we will only look at your mitochondrial DNA.
Okay.
So looking at your all these single nucleotide polymorphisms, and also there are also called length polymorphism that will actually group your mitochondrial DNA into that specific haplogroup. First, we will try to put you into these macro haplogroups, and from there, we will actually search down using specific primers to later put you into these ethnically specific haplogroups.
So it's like a filtering system. You actually put it into big groups and then you filter it down to
correct.
small groups. Oh, okay. All right.
Then later on, what we can do actually from what I did in my lab, so I will actually put your information into a database. So that particular database will actually create a phylogenetic tree, like a tree, where to put you, where are you closest to? Maybe you will be actually, like yourself, maybe you will put into the southern part of India or northern part of India. Okay. Where did you actually come from?
We'll have to take a short break. When we come back, we'll actually talk about the pitfalls of genetic testing, maybe the difference between DNA testing and gene testing and things like that. Stay tuned, we'll be right back. Okay, welcome back. We are talking about genes, DNA testing, and the essence of life with me, Dishen Kumar. And we can move on to another interesting thing that's happening in the US, which is prenatal testing. And they actually, there's a lot of conversation about this. There's a lot of people saying there's a lot of risk, you're not supposed to do this, and things like that. And do we have any companies in Malaysia that actually offer this type of services, or is it quite famous in Malaysia?
I think this prenatal testing is not very famous in Malaysia. Other than we do have this prenatal for this karyotyping to check for whether the number of chromosome of the baby is correct or not. So
Okay.
from there we can actually diagnose whether the baby is actually like a Down syndrome case or something like that.
So that is done when the fetus is
Maybe about 6 months.
6 months.
Yeah,
Okay,
about 6
okay.
months.
So that's not prenatal. That's just like the baby is already formed and
Yes,
exactly.
yes,
Okay.
yeah.
So there's nothing before that when the fetus is still a cell.
Oh no, it's very high.
Yeah.
It's a
Okay.
bit risky. Yeah.
But we have to talk about the ethical question, because that's something that's already been talked about in the United States when they have this prenatal testing. If a couple has this prenatal testing on their unborn child, and they find out the child has a rare genetic disorder and things like that, based on your medical opinion, do you think they should do something about it? Because that raises a very big question, because ignorance is bliss, that they say, So what should be done?
Okay, I'm not quite sure what is actually from my what that I know. If it is being advised by the doctor to actually, the best thing is to get the baby, to get an abortion. So it is allowed to. But as a parent like myself,
Your
if
personal opinion.
Yeah, my personal opinion.
Yeah.
If I'm already 6 months pregnant.
Yeah.
So knowing that my baby having like Down syndrome, so will I carry on with the abortion, or will I carry on with the pregnancy? So for myself personally, I will carry on with my pregnancy, because whatever it is a gift from the God. So I don't think it's right to just terminate a life just like that. So I think it's back to the parents themselves, so whether they want to actually continue with the abortion or carry on with the pregnancy.
Okay, and
It's very difficult to comment on that.
yeah, I know, and because it raises a lot of questions, like if there is something wrong, if I find out there's something wrong with me, if when I do this gene testing and things like that, because you get markers, right? You get markers,
Exactly,
and you know if you have a predisposed condition, a genetic condition, what does one go through? I mean, is there cases where people actually find out, and has it really helped a lot of people, gene testing, to find out whether they have any predisposed disorders and things like that?
Yes. There's a lot of testing currently available in the market, where you can actually check on your tumor markers and then on these cancer markers. There's a lot in the market. You can choose from the racks. But having said that, having a predisposed gene doesn't mean that in like 20 years in future that you will actually having that disease. So like let's say like cancer marker, they have tested, or they have actually looked into the statistic or the frequency of these people having breast cancer with the marker, with the genes. Yeah, so maybe within the patient themselves, they only like 25% of the patient having the genes. So
Having that is not necessarily doesn't mean
that
that you will actually carry the disease in the future.
But doing that test, like you say, the story of Angelina Jolie,
Yeah.
doing that test actually made her aware of what is actually wrong,
Yeah.
and
In her case, because she knows that her auntie is actually does
Have that.
breast cancer. So her case is actually at the very extreme end, where she decided to have double mastectomy. So back to the question whether how do you take that information?
Yeah,
How
yeah.
do you take that information?
Yeah.
And the good thing of knowing that you have a very high risk of having a cancer, the best that you can do is actually to change your lifestyle, to eat healthy, to live healthy, stop smoking, stop drinking. So things like that will actually improve your health, and maybe it will help you to prevent you from getting the disease. Yeah.
Okay. All right. And there is a lot of conversation about testing, genetic testing, and things like that. And in Malaysia, like you said, there is that service, right? To actually talk about genetic testing.
Yes,
not
there's a lot
DNA,
of
not Ancestry, but genetic testing in Malaysia.
Yes, we do have. This we can actually do it in a la carte mode, where you can choose whatever markers that you want to test.
Oh, okay.
That is one thing. And also you can have this, we call it like nutrigenomic studies, where you can actually get to know what are the genes that you have, and what are the best food that you can take.
Oh, okay.
To keep you healthy, to prevent you from getting diabetes.
So it's a dietary plan as well.
Correct, like a dietary plan.
So
but
you actually
from the genetic perspective.
Okay,
Yeah.
okay. So you actually have all that information with you, but at the end of the day, it's still whether the person wants to take that information or not.
Yes, exactly.
Okay.
And other than that, you also can actually have like a full profile of what are the allergens. So what you should avoid.
Okay.
So
what is your body allergic to?
Correct.
And
Not the
a full workup of your whole body
exactly.
from A to Z.
Exactly. So you can choose from what panel you want to have a look. You can have a whole cancer panel you want to test, whatever the genes they have actually found in the cancer cases, and you can have all sets of panels for tumor markers, a set of panels for allergic reactions,
and things like that.
Yeah.
And the works. Okay.
Yes.
All right. Thank you, Prof. We will be back. The next time we're actually talking about gene editing. It's a very interesting topic. Stay tuned. We'll be back after this. Alright, welcome back. We are talking about DNA and our genes and our essence, the essence of life. Okay, and we have Prof. Dr. Zafrina Zainuddin. Prof, we have been having a very interesting conversation. We have to go into a very controversial topic in that sense, which is gene editing.
All right.
So, gene editing, it's been talked about a lot. There's been a lot of conversations. Everyone is talking about it. What does it actually consist of?
Okay, basically it's just a very simple method on to change the sequence of a gene, to change or either to change a single mutation to the one to a better one, or to change the mutation that causes a disease. So for you to obtain the preferred phenotype. So basically there are a lot of methods, but currently CRISPR is one of the in things.
So, right now. Okay.
Yeah.
We have to talk about the conversations. There's been a lot of talk about the gene editing thing because there's a lot of fear.
Exactly.
The fear of the unknown. What are we actually capable of? We now see in movies, people are talking about gene editing. A monkey is as big as a building and things like that. So people don't really understand where we are exactly in that sense. They say we are actually playing God. So what is Prof being in that? Doing that gene editing, what do you say about that?
Okay, theoretically, we have a method. The method has been proven to actually, and the method is capable of changing the DNA sequence. Theoretically, it can happen, but practically, I'm not sure how successful it could be. So, like the Chinese scientists has actually claimed that they have a twin edited gene babies have been born. So. Is it true? So
it's a claim.
It's a claim. So because he has actually violated all the procedures in the lab and everything, the ethical clearance and everything. So we can actually how to validate that, how to make sure that it's true. But theoretically, it is something very scary. If people can actually change a human, a natural, what God has created, do you like to see 10 people having something exactly what you are currently having? So, do you want to see another Usain Bolt, okay, winning an Olympic medal, but knowing him is as a genetically modified organism? Is it fair, socially fair? So, I think there are so many things to actually talk about when we talk about gene editing. But one thing for sure, if we can actually edit the gene that causes the disease, we can actually this genetic engineering is something that is huge. But if we can actually focus into to cure a disease that actually caused by a single gene mutation, that would be great.
Yeah, there has been a lot of talk about doing that. One of the reasons gene testing or actually gene editing.
That was the main.
That was one of the main reasons. Yeah. Like talking about the playing God and things like that. Do you think are we very far from actually creating a human being with genetically modified human being? Are we very far from that? Are we like 50 years behind that, or is it not as far as we think?
Theoretically, no.
Okay.
So because the method is already there, is this how whether it is going to be a positive, whether it's going to give a positive impact or a negative impact. But theoretically, lab-wise, method-wise, the method is already there. The method has been established. So how are we going to use it? Is again is the question, because like what we mentioned just now, CRISPR is actually is a DNA strand. It's a strand that is actually being created by the bacteria to protect itself against the invaded virus. See, so it's for its own protection. But as a human being, we are so creative. So now we are using it to actually change, to make our strawberry more sweeter, and then we are changing our chicken so that we have a bigger chicken, or a chicken that are actually less prone to diseases and everything. So how we use the technology actually.
Okay. And CRISPR is actually quite interesting because when you read about CRISPR, a lot of people talking about it. It's actually there is a memory in that sense. You actually know what viruses actually have.
Based on that short sequences that has been actually kept by the bacteria, so we can actually know what are the virus that has actually attacked that particular cell. That is actually, it's a mechanism of protection.
Okay, all right. And moving on, other uses of CRISPR, we can talk about disease. How does that can be used, like fighting off diseases, or maybe can CRISPR be used or gene editing be used to find a cure for like rare diseases and things like that?
Yeah, I think there's one clinical study. They will actually they have injected these cells that has been modified or engineered using this CRISPR method into human. I think that has been done some time back in 2017, but I'm not sure whether it was successful or not. But that particular study was actually to cure a disease. So there's also one study, I went to one of the conference, that particular presenter, he said he has actually successfully cure a boy, a 4-year-old boy, having this muscular disease where the boy actually, when he gets older, he will actually, all the muscle will get weaker.
I see.
If I'm not mistaken, it was Duchenne muscular dystrophy. So he did this some genetic engineering, and he put it back, the cells, into the boy. And after few months or maybe 1 year, the boy starts walking. So the photo that he showed to us was the boy was actually climbing the stairs from not being able to do anything. So there are the good things from this genetic engineering, but again, how you want to use it.
Okay.
Yeah.
All right. Maybe the final question: what can we actually expect for the future? What actually do you think, maybe in 10 years, 20 years time, that this genetic engineering will actually help?
My fear is actually to those who created this all biological weapon. Yeah, that is my fear, where people abuse the knowledge, the method and everything to create something that is can cause disaster. But for curing disease, I can see that there's a lot of potential that we can cure this beta thalassemia that is actually caused by a single mutation in the gene. Because we have a lot of cases of thalassemia in Malaysia. I have 2 cousins, they die at a very early age because of thalassemia as well. So precaution is good. We have all this screening, all this gene screening, right, for thalassemia. But in future, like in 20 or 30 years, if you can go to a clinic and give your blood or your cells for a genetic engineering, and after that, after a year, you are cured, that is a miracle. That is what we are actually looking forward to, rather than. But my fear is like there are more people actually abusing the technology rather than actually doing it for good.
But all we can do is just hope for the best.
Yeah, hope for the best.
All right. Thank you, Prof, for joining me today, and I appreciate everything that you've said. If you want to watch this back, please follow us on all our social media platforms and also the Ashrawani app. My name is Dishen Kumar. Thank you for watching. Goodbye.